Let's continue talking about some types of inheritance that are different from simple Mendelian patterns of genetics. So for this lesson, we're going to be looking at scenarios and examples when a gene produces multiple phenotypes. Most genes do have multiple phenotypic effects on an organism, and this is a phenomenon called pleiotropy. One example of pleiotropy is with sickle cell anemia. A person with sickle cell anemia has sickled or crescent-shaped red blood cells in contrast to a normal round red blood cell. Now, the sickled cells themselves is a phenotypic effect, the shape of the cell being different, caused by a mutation in a gene. But there's all sorts of symptoms that a person might have that has sickle cell anemia. So each of these symptoms or health related concerns that are down in the purplish looking boxes there, these are all also phenotypic effects. So because it's not just the fact that the cells are going to be misshapen, but that it leads to all these other causes, all these other phenotypes and symptoms, is why this is a type of pleiotropy. Oops, didn't mean to go back. Sorry about that. Okay, and along these lines, there are also some scenarios with inheritance where there's more than one gene that determines a phenotype. And we're going to look at two types of this, epistasis and polygenic inheritance. First, with epistasis. In many cases, a gene at one locus, or if you recall, that means location of a gene on a chromosome, affects the expression of a gene at a different locus. This is known as epistasis. And to hopefully give you a better understanding of this, we're going to work through an example first of more of an analogy, and then how that relates to a true epistasis example in humans. Okay, so imagine you have this assembly line of people. Each person labeled A, B, and C has a particular job to do in the course of this process, with the output or the result being a blue circle being painted. Person A's job is to get the white paint, put it in the paint tray for person B, who adds the blue color, like the tint at the local home fixer-upper store. You know, you get the base color, and then they add the tint based on what color you want. And then you take that can of paint home and you paint with it. And so person C, their instructions is to take whatever paint is provided to them and draw a circle. So like what we're seeing with inheritance, there could be different versions of this process. So maybe instead of person B adding the blue tint to the white paint, they add red tint. They're still, the operation of the process is still the same. Person A still gets white paint. Person B now is adding something, but it's red instead of blue. And person C is still drawing a circle, except now the output is a red circle. Okay, so this is getting at the idea that genes with epistatic relationships tend to code for proteins that work together. Okay, so imagine instead of these people, that those are proteins. Each protein has a particular job, and ultimately the goal is to cause a phenotype that is a result of the three genes working together. And we'll come to an example in a few minutes. Okay, but we're not quite at the full concept of epistasis yet, but we're getting at it because of this idea of the relationship between these proteins that are working together. So let's continue on and get there, hopefully soon. Now imagine how, what would happen if one of those persons involved or one of those proteins involved was broken or didn't get the full message, didn't do their job. So in this scenario, A is broken. A doesn't know what to do. They're just kind of stuck. Although B and C are ready to do their part, there can't be any output because A wasn't there. A didn't show up to work that day. So let's try a different version of this and see how that might affect the output. If C isn't functioning properly, even though A has the white paint, B adds the red tint, C doesn't know what to do. So again, there's no output. Even though the output is again a blank poster, nothing was painted, the cause was different than when A was broken. So because because one of the three involved in the process was not doing their part, there could be no output. So it only takes one faulty gene to disrupt the process and result in a different output, or no output at all in this case. But now let's take a look at a true example of epistasis. It's also an example of incomplete dominance, and that's human eye color. Now we're going to simplify this because, as you might know, human eye color is more than just these two options of brown and blue. We're going to think of it more as pigment versus no pigment, but because there are many different varieties of eye color, it's actually a type of incomplete dominance that explains that phenomenon. But for the purposes of this and the understanding of epistasis, just think about pigment being anything but blue eyes. And blue eyes are actually lacking pigment in the iris. So eye color is determined by about 16 genes. It's pretty complex. But we're going to simplify it here to the two that are mostly responsible for it. And they are abbreviated here as HERC2 and OCA2. So HERC2 and OCA2 are both involved in the production and expression of pigments that result in the pigmentation of the eye. Varying degrees of melanin is actually what results in the pigmentation of the iris. And so again, these are two proteins that are working together to result in the production and deposit of that pigment on the iris. So as you can see in the first scenario, if both of those proteins, HERC2 and OCA2, are working properly together, then there will be pigment of the iris. But if either of them is not working, is faulty, or not coding correctly, then there won't be pigment. Oops. And that person will have blue eyes instead of brown pigmented eyes. It's actually very complex. If you'd like to read about it in the notes of the slide, if you download the PowerPoint, you'll see a link to the article and a more deeper explanation about how eye color works in humans. So some of our traits are pretty complex. It's not just a simple Punnett square that we could do to predict the outcome of a cross for certain traits that humans have, like eye color and also skin color. Skin color is another type of inheritance that's determined by more than one gene, and this is called polygenic inheritance because there's an additive effect of these two or more genes on the single phenotype, which is your skin color. So what makes this different from epistasis, which was when you had the genes working together to produce a phenotypic result, here the phenotypes appear across a spectrum. So for the skin color, for example, some people have really, really light skin and some people have really, really dark skin. And if we plotted every single person on the planet's skin tone on this graph, it would actually have this bell-shaped curve to it. Most of the people in the population are somewhere in the middle, having moderately pigmented skin. The lowest numbers are at the very, very light and very, very dark end of the spectrum. So it has a measurable effect on the population distribution. It's quantitative in terms of its variation. So why is that? Well, it has to do with the fact, again, that it's coded by multiple genes. This is a simplification of that, showing only 3 genes that control skin color, A, B, and C, but in fact there are more than just 3. But what I want to show you here is what a possible Punnett square would look like if you had 3 genes that you're crossing together. We've done it with 2 for the Mendel law of independent assortment, but we're not going to have to do this, don't worry, but this is a massive Punnett square, 8 by 8 with 64 possible outcomes. To show you what would happen if the lightest skin tone, a person with the very, very lightest skin, which would be recessive for all 3 of those genes, had children with a person that had the very darkest skin and had the dominant homozygous genotypes for each of those genes. All of their children in the F1 generation would be heterozygous for those 3 genes. Capital A, lowercase a, capital B, lowercase b, capital C, lowercase c. So this would be a trihybrid cross, and the resulting phenotypic ratio at the bottom would be the 1 out of 64 to 6 out of 64 to 15 out of 64 to 20 out of 64, and then back down again the same way, showing that again the middle, the median skin color is the one that is the most prevalent. So that Punnett square is what is applied as a predictive tool for the human population, assuming that all of these skin tones are present in the human population. But on the note about skin color, also bear in mind that environmental factors such as sun exposure can also influence skin color. So some of our traits are inherited, but can be altered because of our exposure to the environment around us. But those changes in our skin color because of the environment wouldn't necessarily be passed to our offspring unless it's causing a change in the DNA.